And thank you very much for giving me the opportunity to share some of our work. I will focus a little bit more on the osteocyte Lacuno canalicular network. But first, thanks to the people involved in this work, so primarily my group members, but also excellent collaborations at the ESRF, both with the team at ID13 and ID16A. So thank you very much already now. So we've seen this before in both Peter's presentations and also by Tillman. Bone has this very complex structure, and as remarked upon by Peter, there is this intriguing cellular network formed by the osteocytes that we would like to understand better, because most of, say, the biological knowledge we have about it actually comes from cell culture and less from what actually happens inside the actual bone. And since it's structured across many length scales, there's a complication in actually doing this in practice. So we've been working over a number of years at various different synchrotrons at sort of two different sets of length scales, and I give two examples of this. One is this intermediate length scale you can see in the middle plot, where we have voxel sizes maybe of the order of, say, 300, 200, 300 nanometers or so, giving submicron resolutions, which allows us to map the osteocyte lacunae that you can see in green, and of course also blood vessels and whatnot, as marked here in red. The advantage of this length scale is that we can do biology. Also something Peter mentioned in his talk, it's really important that we arrive at a stage where we can do, say, less physics chemistry experiments and more biology experiments, which requires that we can measure very many samples. So we measured, for instance, of the order of 200 samples in one experiment, and this actually works well. But of course, you have to think about data handling and sample exchange rates. And then I'll refer to a little bit later to how we can actually measure the Lacuno canalicular network by higher resolution tomography. So here's a simple example. This is from rodent bone. As you can see on the right, all the little orange guys, these are osteocyte lacunae, and rodents are quite different from men and women, as it turns out, and from humans. So the way the bone is formed is quite different. So you see here these light orange circles or blobs. These are osteocyte lacunae from what's called endochondrally derived bone, whereas the darker orange ones are formed by apposition. So by measuring large volumes, we can actually map out the impact of how the bone is formed in the orientation distributions of the osteocyte lacunae, as an example here. So as one question that we've addressed at the ESRF with this is looking at a process that has been proposed for these osteocytes, which is their ability to lyse, or that means break down the surrounding bone and thereby contribute to calcium homeostasis. So when your body needs lots of calcium, it has been proposed that the osteocytes could directly contribute to this. So there have been several models in which this has been proposed. One would be immobilization, and there we did not see it in rats. But if you really need a lot of calcium, for instance, if you are lactating, and especially if you are a mouse and you lactate many mouse children, then you would expect really to have to mobilize lots of calcium. And so in CD1 mice and in C57BL6 mice, this has been demonstrated. So we thought it would be a perfect opportunity to do this with three-dimensional techniques. And of course, we, being a bit naive, we studied a different mouse strain in LMDI mouse. So we did holotomography at ID16A with a voxel size of 130 nanometers, giving a resolution of roughly 326 nanometers. And as you can see in panel A, this gives us very nice delineation of the yellow osteocyte lacunae. But sadly, the lacuna volumes did not change between the lactating group and the non-lactating group. We verified this by lacunae area measurements using backscatter electron imaging and light microscopy. And this led us to the conclusion that there is no real sign of osteocytic osteolysis in this mouse strain. And so what we're now hoping to do is to, of course, compare with other mouse strains and see to which degree this is really a general physiological response or just related to a few mouse strains. Sorry. So to obtain higher resolution, in order to unravel the full cellular communication network, we need higher resolution, and one way to do this is to do phase propagation holotomography, as implemented in IND 16A. And we and others have done this. I will just show one result of this, done with 50 nanometer voxels. So what you can see here on the left are two cells from a large volume. So here is an osteocyte lacuna in number 1 and number 2, and you can really see they're interconnected by this vast network that also Peter alluded to in his talk. Now, having these data in 3D allowed us really to see many new features. And on the right, you can see there's a little blob moving around over here in this single cell. And so when we were looking at this more, we saw that there were these blobs within the network that we call junctions. They occur in the places where several canaliculi cross. And that then led us to think about how, which are the roles that this Lacuno canalicular network is believed to play. So one of the proposed roles is that these cells sense stress induced by fluid flow through these canaliculi. And so if we were to have a void inside this network, that would of course change the fluid flow dynamics. So here comes this idea of correlative imaging, also proposed by Peter, because we need now to know if this void space that we see is really linked to the fluid flow network. So inspired by the work of Peter, we conducted the same experiment he showed before, using confocal optical microscopy. And we could see that in several different animals, we observed these junctions, and they're connected to the fluid network. So that means that they for sure impact fluid dynamics within the osteocyte lacuna canalicular network. So we proposed two roles for them. One would be that they act as fluid flow modulators with an impact then on the stress sensing ability of the cells. And another one could be that they could act sort of as highway junctions, allowing for interconnections for multicellular communication. And of course, we would need to do further experiments in order to prove this. So that told us a lot about the osteocyte lacunicular network itself, but it does not tell us so much about the surrounding mineral. And so to do this, we would use diffraction. As a first example, I would just show you a larger scale experiment, and then we will increase resolution to go closer to the osteocyte lacunae themselves. So in this experiment, we looked at osteons, and osteons, they form by the carving of a tunnel, which is then filled from the outside in. So we can follow the biomineralization history by spatially resolving the mineralization profile across the osteon. That's of course needs to be done with submicron resolution in 3D. And we did this particular experiment was at Petra, but of course the same can be done at the ESRF. This was done with a 400 nanometer X-ray beam. And to cut a long story short, we could look at the peak broadening of the diffraction peaks, both corresponding to the broadening along the z-axis and perpendicular to the z-axis. And in both cases, we could see the crystallites close to the osteon canal here and here. They were smaller or had broader diffraction peaks, reflecting that the crystallites most likely are smaller. So why can this be? Well, close to the canal, we know that mineral modulating proteins such as osteopontin play an important role in the final stages of the mineralization process. And in this paper referenced at the bottom here, we show that if you remove the ability to properly process osteopontin, all this region here with the smaller minerals is actually not mineralized at all, because mineralization is completely inhibited. So this shows that the variations or modulations in the non-collagenous proteins have a very strong impact on the local properties of the mineral within. Knowing that the protein such as osteopontin is also upregulated close to the osteocyte network. We then thought we would really like to be able to obtain the same information, but at a much higher resolution so that we could study the periosteocytic mineral. And so to do this, we would do the tomography using X-ray diffraction and X-ray fluorescence. So this we did at ID 13, and here's the first pilot experiment that we conducted with a beam defined by multi-layer Lauer lenses, 12 kilovolts, focused onto a small bone sample, roughly 3 microns across, that you can see in this SEM image in the center. And then we measured the diffraction and the fluorescence signal as a function of orientation. And let's say this was a rather heroic experiment because we had to sort of do each orientational slice by hand. So it took a little bit longer than we first had hoped, but in the end, it worked very well. So we measured 92 slices that allow us to do tomographic reconstruction. And the image on the right there shows you a raw projection image of calcium fluorescence. And now if we reconstruct this tomographically, I'll show you a slice through the data on the right here, and you can see the void spaces, and we can also see fiducials from the gallium that we. Sorry.
Henrik, it might be my phone, or it might be, but you were breaking up. But maybe please continue. Okay. Maybe it's only me.
Okay. I'll try to continue.
Yeah, it's fine, Henrik. Thanks.
Yeah. Okay.
Okay. Sorry.
Okay. So we prepared the sample by focused ion beam milling, and that leaves some gallium on the surface of the sample that you can see then reconstructed on the right. We look at this then allowed us also to look at low calcium regions, which delineate most likely the canalicular network, as you can see here in blue. If we then look at the diffraction signal, we can see that that we treated by Rietveld refinement. So the amount of crystals is given by the crystallographic scale factor, and we can see a very good correspondence between the calcium signal from fluorescence and the local mineral crystal content as determined by diffraction. Now, if we look at the individual diffraction patterns, we can see that within the sample, these are these two here, we get okay looking diffraction patterns with a fairly low background. On the surface of the sample, there's a very large background. And that's because of this gallium fiducial marker, gallium being an amorphous background generating sample. So from this, we could show that we could do combined XRD, XRF, CT on bone with a better than 120 nanometer 3D resolution as determined by edge fitting to the data. Okay, so this demonstrated that the experiment is possible, but didn't teach us a lot about the sample. So what we're doing right now is trying to do this experiment again, even better. So we've carved out, as you can see here, a newly formed osteocyte sitting right here. And so we prepared a bone specimen just close to the osteocyte. And we've looked at it in two orientations. So one this way, and then at 45 degrees. And I think I will just skip the left part here and focus only on the right part that shows you that the calcium content is basically the same in these two orientations. If we now look at the diffraction signal, we can see that we can measure the orientation of the crystallites by using fits to the azimuthal intensity dependence, as done also in Peter's work from small angle scattering. And we can now then look at the orientation of the crystallites as a function of distance from the osteocyte lacuna. So on the top, I show you the orientation as determined by small angle X-ray scattering. The color gives you the angle, and the intensity of the color gives you the degree of orientation. So what we can see is that there's a fair degree of alignment from the small angle scattering all over, but systematic variations with distance from the osteocyte, which is at the top. And the whole image across is just 4 microns. And at the bottom, we can see that in one orientation, we see a more or less uniform but rather low C-axis signal, corresponding to that the crystals are predominantly parallel to the beam. But when we turn the sample 45 degrees, we see that there's this patch, roughly 2 microns around the osteocyte lacuna, where there's a very distinct crystal orientation. So using these nanometric X-ray beams, we begin to be able to actually unravel the fine details in the periosteocytic. So with this, I would like to conclude. I hope I've shown you that the X-rays are fantastic, but I guess here I'm preaching to the choir. We used tomography to show that MDI mice probably do not display osteocytic osteolysis. We unraveled canalicular junctions using nano-holotomography and demonstrated that it's possible to do XRF, XRD, CT at a very high resolution. And this, we hope, will allow us to map orientation of mineral and nanostructures close to the osteocytes. And we're working on this now. And so with this, I would like to thank you very much for your attention and entertain any questions.
Thank you very much, Henrik, for this very interesting presentation and where the limits of resolution could go. Are there questions? There's no open question right now. Then maybe I might start with my first question. Imagine we could do something like laminography on laterally extended samples, but with a similar thickness to the ones treated right now. Would this asymmetric extension of the field of view be of value? Should we try hard to do this? Or would you say it's a waste of time, rather try to increase the overall 3D volume? Because it's very different, let's say, instrumental efforts to do the two, and as well in terms of the algorithm.
I mean, it's always dangerous to ask users if they would like both ice cream and cake, right? Because the answer will always be yes. But so there are two challenges. One is we are reaching length scales where sample preparation really becomes also a very significant challenge. So the ability to work on samples that are larger, at least in one dimension, would be welcome. I would have to think a little bit more about it if I have to say I would prefer one over the other. A significant challenge is also total dose on the specimen. I mean, everything becomes a little bit fried after a while in the beam, and I think this is what's actually limiting the degree of observation that we can do, certainly with the nanoscale beams, because the flux densities become really quite high. Mm-hmm. So one would have to take sample damage very carefully into consideration.